Hi everyone, and thank you for having me. So I will be taking you on this little conceptual journey talking about between species genetic interactions. And okay, it is very faint. I hope you can read the slides. So this is basically the product of the first year of my PhD, where I was reading about these different kinds of interactions. I was reading up on gene-for-gene interactions, allele matching, genotype-specific interactions, and so on. And my questions very much was, okay, how do I classify something, or how do I define when something fits in where? Especially since we already heard in the introduction that the classification can depend on different things, I guess the biological nature in one thing, but then also maybe at what time we look at it, but also, I guess, the resolution of the genetic data we have available. And I'm stealing the same examples as Ben did. So here we have this Daphnia system. And then originally this was described as having genotype-specific interactions, so depending on the host genotype and the bacterial parasite genotype, the outcome of infection would be different. And then later on this was refined to have like this specific allele matching mechanism. So depending on what data we have available to us, we can classify things differently. But then also there's field-specific data, like if I want to think about gene-for-gene interactions, I will definitely find papers about it in these crop pathogen systems. But does this mean that we only have gene-for-gene interactions in these systems, and do all of these systems only have gene-for-gene interactions? I doubt it, but I think this bias might also impact the way we ask questions, what kind of answers we are looking for. So my question very much was, how can I find, I guess, some sort of unifying framework wherein these concepts still exist, of course, but maybe I'm able to also describe things that lie outside of them, and just something that helps me describe things in a dynamical manner. So being able to switch between these different things in a way that does not impact how I think about the system. So I guess this is one approach I did first was like, okay, how do I structure this? So by thinking about these things as subsets of each other, so having based on their specificity. So having interactions that are based on a genotype level, things that are mediated by specific genes, and then things are mediated by specific alleles. And then the question is, so what is the overarching thing? Well, we went with the idea of calling it endogenomic epistasis. And the epistasis bit refers to the fact that we have interactions between different genes, and the endogenomic bit refers to the fact that these genes happen to lie in different genomes, so genomes of different species. Meaning that what is happening in the genome of one species, and depending on what is happening in the genome of another species, that will have different effects. And I guess you can also think about this in the context of the extended phenotype. This is not just a term we picked out of thin air, but instead this is something different people have used for their empirical studies. So they use the term endogenomic epistasis to describe between species genetic interactions. But my question was more on the conceptual mathematical side. So how can I find a mathematical formalism to describe this, to characterize these interactions, to be able to compare them and describe different systems? So first, just to nail what I mean with endogenomic epistasis, I'm showing you this extremely simple fitness landscape, where on the x-axis I have two different genotypes, 0 and 1, so reference genotype, and then the presence of a genetic variant. The genetic variant can be whatever you want it to be. It can be a single, like a SNP. It can be presence absence of a whole gene of a haplotype. But then the idea is that for each genotype, focal genotype of the species I'm studying, I can measure the fitness, and then the mutation can impact this fitness. But then also for the same genotype of my focal species, I might have different levels of fitness. And of course, we can think about developmental effects, environmental effects, and so on, but this is very much in the context of species interactions. Right? So the idea is that depending on what is happening in the genome of the species I'm interacting with, so like a host pathogen, depending on which host genotype I'm infecting, I will have different levels of fitness. And this difference of fitness is already what we consider endogenomic epistasis, because we have an effect of the genes in the other species. And to make this slightly more complicated, but still the same, so now I have the focal genome in orange, and within the focal genome, I'm looking at two different genetic sites, so 1 and 2. And then I have a single partner genome with sites 3 and 4 in purple. And of course, we could extend this to many more species and many more sites, but for simplicity, we will just stick with 2. And then again, at each site, I'm just looking at presence absence of a genetic variant. And the idea would be that for each genotype of the focal species, I would measure fitness. So focal fitness would be the product or would be a function of my focal genotype and then the partner genotype that are interacting. And so you'd measure fitness, you have some sort of baseline fitness, but then of course, fitness can deviate depending on different effects, right? So these are all possible effects that are possible in the system. In red, in the red arrows, they show you the additive effects, meaning, okay, I now have a mutation, this changes fitness. But then we can also have interactions between different sites, right? So we can have classical epistasis, so in like epistasis within the focal species, but then we can also have all of these things that contribute to endogenomic epistasis. So this can be interactions between both genomes, so these pairwise endogenomic effects, then high-ordered endogenomic effects, and so on. So basically, as soon as the genome of the other species has an effect on the focal species, this is endogenomic epistasis. And then these are all possible terms that we might find or that we could use to describe different kinds of interactions. And how we would apply this is something like this. So again, in keeping with so on the x-axis, I have the different genotypes. In the top row, I have the focal genotype of the species I'm interested in. And then in the bottom row, I have the partner genotypes. And then I'm just combining things. The light gray means absence of any genetic variant, so like wild type. And then the dark gray shows us the presence of a mutation, for example. So I would start with wild type, and this gives us like the expected baseline fitness that I'm measuring. And then the idea is that I go through all of these genotype combinations and see what happens. So here I'm showing you, I guess, a classical gene-for-gene interaction. And then we would find something like this, where here we have, I guess, the matching of, like, in the classical way of the resistance gene in one species and the Abrils gene in the other species. So matching of gene 1 to gene 3 in the other genome. And this decreases fitness. Then I go through this again, and I might find there's another resistance gene matching to a different avirulence gene. Then this gives us this fitness pattern. So what does this look like mathematically? Using all of these terms that I explained to you previously. The blue terms are pairwise epistatic interactions. These are the ones that confer the matching of the genes. And then really cool, we can already see that there's more going on in the system. So we have this higher order epistatic term right at the end in purple, which is basically the masking effects of multiple resistance genes. Since gene-for-gene interactions are this like we think about them as a quantitative interaction, having multiple resistance gene doesn't make a difference versus just having a single resistance gene. So here we already have like this mechanistic insight on what is happening due to this higher order effect. And the cool thing here is that now if we have some sort of deviation of this pattern, we can simply change the terms, add terms, or take away different terms mathematically. And this would give us a different kind of definition. So to go over this, how would I formalize between species genetic interactions? You would isolate different genotypes. You would combine them and measure the fitness or any kind of phenotype you're interested in. You would then infer the mathematical terms. And as I said, the cool thing here is that we don't just have an accurate description of what is happening, but we also already have a mechanistic insight. This is also something that we can then change over time. So this is something that we could use to describe and classify the nature of between species genetic interactions, then also classify them again in different ways. And we can always go back and refine depending on what data we find or what kind of information we have on the genetic level. And since this is also about coevolution, I will give you some teasers about how we can use endogenomic epistasis to think about coevolution. I unfortunately don't have time to go very in-depth into this, but what I presented so far is how to use endogenomic epistasis as a more descriptive way, but we can also use it as a framework to think about, well, coevolution happening. So different things that we could borrow from the field of epistasis and apply them to the study of coevolution. So one of the things I would like to think about is like having like cross-species historical contingencies, navigating fitness landscapes of both species together, and how this can affect the ruggedness of a fitness landscape, for example, the presence, absence of different species, and how this might constrain evolutionary trajectories of single species that are walking along this fitness landscape. And I'm finishing early. I think I was very nervous and I skipped a few things, I'm sorry. But I'm already at the end of my talk, and I would like to thank everybody for their attention, and my lab back in Bern, and my co-supervisor. Yeah. Yes, check out the preprint. There's a QR code.
Okay, again, my name is Abbey Ramirez, and I am in Amanda Gibson's lab at the University of Virginia. And today I'm going to be talking about one of my PhD projects, temperature alters genetic specificity in a host-parasite interaction. Okay, so one of a really prominent idea is that host-parasite interactions can maintain genetic diversity through negative frequency-dependent selection. And I have that depicted here, where adaptation to a common host, first we'll do host 1, adaptation of the parasite population to host 1, I'll say, will give a rare host an advantage. So as this increases in frequency, its compatible parasite population comes with it, thus resulting in a fitness advantage for the rare host in the population. So the underlying mechanism of this model is the matching alleles model. And this assumes strong genetic specificity that throughout this presentation I will be referring to as G by G. So I have a really simple matrix here that is really similar to the one that they had in the intro presentation, where host 1 and parasite A are compatible, and host 2 and parasite B are compatible, and only these two combinations will result in infection. This also assumes that this specificity remains stable. This has been one of the biggest criticisms about this model because it's thought that it could possibly change under certain conditions, such as changing environments. So this stability is tested by a three-way interaction that I will be referring to by G by E, or genotype by environment. And there's only 3 studies that I could find that test for this interaction, and the results of these studies are conflicting. And it's been hypothesized that the lack of these studies could be due to the amount of statistical power needed to actually detect a three-way interaction. So there's a couple ways that I can imagine a G by E happening. So we're going to return to our initial 2 by 2 matrix here. And if we have an environmental change, any environmental change, it's possible that the rank order of the compatibility can change. So you can see that it completely flips here, where now host 1 is no longer compatible with parasite A, but it is with B, and then vice versa for host 2. So the other way that I think this could happen, if we return to the same matrix and we have an environmental change, now all of a sudden we see that every single combination is going to result in infection. Both of these circumstances could result to where we lose that rare host advantage, and then we could either see genotypes in the population going to fixation or losing genotypes in the population. So I set out to test if genetic specificity is robust to environmental variation, and I will be using temperature as my environmental variable. It continually changes, and it is really important and changes quite a bit during my study organism's reproductive season. So for my study system, I am using a crop pest, Meloidogyne arenaria, as my host. And its parasite, which is an endospore-forming bacteria, Pasteurella penetrans, and this infection looks like this, which I think is probably pretty hard to see probably from there. But as the nematode moves through the soil, you can see there's like these little bumps here. Those are the endospores that the host is picking up while it travels through the soil. So this is a really good evolutionary model system for multiple reasons. We have indication that there is genetic specificity in the system. And I think it was mentioned in the last two talks, the Pasteurella ramosa and Daphnia. Pasteurella ramosa is related to Pasteurella penetrans. So we kind of have that suggestion that it could also be following the matching alleles model. We also can expect really strong selection in this system for two reasons. First, the parasite is obligate to its host, so it can't reproduce outside of it. And secondly, it does sterilize the host. So to obtain host lines and parasite sources, we collected individual plants for the host and soil to get the parasite down in some infected USDA fields in Tifton, Georgia. The host is purely asexual, so it's pretty easy to come back to the lab to establish clonal host lines in the greenhouse. And then to gather my parasite sources, we can just mix soil and water, allow for like the heavier sediment to settle to the bottom, and then just pour off the top, which will give us these spore suspensions that we can use for our experiment. So for my experimental design, it was a pretty big experiment, so I have only modeled a single host here at 1 temperature, but overall we did 6 host lines. So I repeated this setup for all 6 host lines. I paired them with 5 parasite sources, and then we did this over the 3 temperatures, 25, 30, and 35 degrees Celsius. And then for every combination, I had 3 replicates, and then within those replicates, we allowed the host and parasite to be exposed to each other for 48 hours. Okay, so assessing affection, we use attachment rate, which is the really standard way to assess compatibility between the host and the parasite. That is just your infected number of hosts over the total amount of hosts in your sample. And we use the first 50 nematodes that we encountered to estimate this attachment rate for every combination. So I counted over 7,000 nematodes and 78,000 spores during the course of this experiment, and I modeled this attachment rate as a function of all of the main effects, so temperature, host, parasite, and then all of our interactions, so our two-way interactions, host parasite, host temperature, and parasite temperature, and then of course the 3-way interaction that we're really interested in, the host parasite temperature interaction. Because this talk is only 12 minutes, I wanted to focus on the 2 that this project really set out to investigate, which is going to be our temperature and our 3-way interaction, the host parasite temperature. Okay, so first we're going to look at the main effective temperature, and before I show you these violin plots, I want to get you oriented. So we have attachment rate on the Y from 0 to 1, and then I'm going to be showing you violin plots of the low, medium, and high temperatures. So for low, you can see there's a ton of variation from very, very little, I mean near 0 to close to 100% infection. This makes sense because this is every single combination that we have in the experiment, so we would expect to see a lot of variation. It gets interesting here between medium and high, because you can see that medium looks very similar to low, where there's a ton of variation, but all of a sudden you can see this collapse in variation at high temperatures, where now more of these combinations are resulting in really high infection rates or attachment rates compared to all the variation we're seeing in low and medium temperatures. Statistically, there is a significant host by parasite by temperature interaction. I'm going to warn you, this is all of the data. It's kind of hard to extrapolate anything from this graph, at least I think so. So I'm going to break it down for you, but first I want to show you the entire thing. So I'm going to have low, medium, and high temperatures, host 1 through 6 over here on the Y, so the rows, and then the parasites will be the columns. Attachment rate is going to be anywhere from light blue to dark blue, which represents low attachment rate to high attachment rate. So for the low, and I want to point out real quick that these are just combinations that did not have enough nematodes in it to get anything. But you can still see the genetic specificity that we've already seen in the system here, where you can't predict the infection outcome based on host or parasite alone. For example, for host 6, the highest attachment rate is with parasite source 5, and the lowest is with 2. And then if you go here to host 2, you can see the highest one is 3, and the lowest one is 1. So for the medium and high temperatures, again, it looks really chaotic. It's really hard to see any changes as you go from low to high temperatures. But what you can see is that attachment rates are getting a lot higher in this higher temperature, which we see in that main effect as well. Okay, so to break this down, I just looked at attachment rate changes from low to high. So I have 25 different combinations here that show the differences in attachment rate. And out of those 25, we have 21 of these that are either increasing in attachment rate like this, or remaining pretty straight. But the ones that have no change initially had really high attachment rates at the beginning, so it makes sense that they're really not able to go any higher. The alternative to that is we do have these 4 that are showing declines from low to high, but those are relatively low decreases. So overall, we're seeing a reduction in specificity or a collapse of variation within this data. So the question, are G by G interactions robust to environmental variation? I hope I made it clear that no, they are not. We do see a significant host by parasite by temperature interaction, and we think that the reduction in specificity at higher temperatures could reduce the rare host advantage that we would expect to see, meaning that either the common host can gain an advantage or the rare host is going to lose their advantage, which could result in loss of genotypes. So with that, I want to thank my lab, all my funding sources, committee members, all my lab and field help, and you guys for sitting here and listening, and I will take any questions.
Oh, did it? Okay, well, I better get going then. Well, hi everyone. My name is Sam Hulse. I'm a postdoc with Emmy Bruns at the University of Maryland, and today I'm going to talk about some of our ongoing modeling work in the lab. So when we talk about transmission modes, many pathogens rely on multiple transmission modes, such as vector transmission, wind transmission. There's a whole suite of possible transmission modes. And this can be a really important factor in understanding both host and pathogen persistence, as well as landscape level transmission and all sorts of important ecological factors. And another important thing is that oftentimes pathogens have a sexual transmission mode. And an interesting distinction about this is that sexual transmission generally only works about infecting adults. So for instance, pollinator transmission is a form of sexual transmission, but juvenile plants don't have flowers yet, so this form of transmission doesn't really hurt them so much. So there's this clear tie-in between age and transmission mode, and an interesting consequence of this is that if you have pathogens that are affecting their host at maybe different parts or through different physiological pathways, that means that the mechanism of both resistance for the host and infectivity for the pathogen might be different and therefore might be under different evolutionary or selective pressures. So a little bit on what motivated this. In the Bruns lab, we work on this system. We study a flowering plant in the carnation family of the genus Silene, and it gets this fungal pathogen called anther smut in the Microbacterium genus. And so on the left, you can see an infected plant. So it's this, if you look at the flower there, you can see it's kind of brown and ugly looking. And so that is the fungal spores here. And so it's a systematic infection that manifests in the flower. and then it can then be transmitted by pollinators to other adult flowers, and once a plant is infected, it results in full sterilization, but without any apparent induced mortality. But we also know that juveniles can get infected just through wind dispersal of the spores. Another really interesting thing is that juveniles are way more susceptible. It can be like 10 times more, juveniles can be 10 times more likely to get infected given transmission than adults. And some other work from our lab, so this is the work of our now sadly former postdoc, well, it's not that sad, he got a job, Sam Slowinski, and a talented undergrad, Irina Lee. And so they looked at the differences in relative juvenile infectivity to adult infectivity across several different strains of anther smut. And so they found this kind of cross-relationship where there seems to be a trade-off in each pathogen strain's ability to infect adults and juveniles. And another thing that we know in this system is that on the host side of things, it seems that selection for either juvenile resistance or adult resistance can be independently evolved. So it's possible to think that hosts have to allocate resources between multiple forms of resistance that either work at the juvenile or adult stage. So here we have another possible trade-off set up. So really, infection is a property of both the host and the pathogen. So this is a quintessentially coevolutionary question. And so we sought out to ask, what are the factors that determine, first, how the host allocates resources between juvenile and adult resistance, and likewise, how does the pathogen allocate resources between juvenile and adult infectivity? And so when you have this link between age and transmission modes, what are the demographic or evolutionary factors that allow you to maintain multiple transmission modes simultaneously? So to answer this question, we developed a model. So for those of you who have seen these kind of compartmental models before, this is an SI model. So we split up our population into susceptible individuals and infected individuals. The susceptibles are given by the green boxes at the top. We have SJ for susceptible juveniles and SA for susceptible adults. And susceptible juveniles mature into susceptible adults at rate M, and then they can both become infected by adult infected individuals given by IA. So if a susceptible juvenile is infected, then it goes into the infected juvenile class, which can then mature into an infectious infected adult. And so we have transmission is density dependent, where we have these parameters beta J for the juvenile transmission and beta A for the adult transmission. And so for those of you that like this stuff, this is what the model looks like written out as a differential equation. I'm not going to linger on this for too long, but yeah, we got math. So we break down our beta J and beta A terms further. So here's a little small, but you can see beta J is given by HJ, which is the host's contribution to infectivity or resistance, and PJ, which is the pathogen's infectivity, and beta 0, which is the baseline level of infectivity. So here, our hosts want to minimize the H's and the pathogens want to maximize the P's. And then we assume that there is a trade-off for the host between adult resistance and juvenile resistance. So that's given at the bottom where we have HA is 1 over Z times HJ. And this parameter Z just allows us to have some sort of adult resistance bias. So I mentioned that in our system, and in many other systems, you see that adults tend to be way more resistant at baseline. So when we have Z is equal to 1, that is where this blue line looks here. That's when basically evolution can optimize for juvenile or adult resistance with equal effectiveness. And as Z increases, then adults basically have a higher level of baseline resistance. And so then we also assume an equivalent trade-off on the pathogen side, where we have the adult infectivity is the reciprocal of the juvenile infectivity. And so these are both, for those of you who've worked with adaptive dynamics, these are accelerating cost curves, which tend to produce single optimal strategies instead of branching. So then to put this model into an evolutionary context, we use adaptive dynamics. So adaptive dynamics is a framework where you basically assume that ecological time and evolutionary time are separate. So when we start out, we have some initial genotypes for our host and pathogen, and you let them reach what's called an ecological equilibrium. So they reach an equilibrium prevalence of the host and the pathogen. And once they reach this equilibrium, we introduce mutations for both the host and the pathogen. So we assume that these new introduced mutants differ from their progenitors by a small phenotypic value that's given by our trade-off curves. And with those new mutants introduced, we then run the simulation again to see if those mutants can successfully invade, or maybe the residence strategy wins out. And so in the end, it basically looks like this figure on the right, where the blue represents the host genotype's evolution over time, and the green represents the pathogen's evolution over time. I'm going to come back to this figure, so don't worry about it too much. But the x-axis is evolutionary time, and the y-axis is kind of the phenotype. And so you can see it's kind of equilibrating at the end, so you reach an ultimate evolutionary equilibrium. So with this framework, we ran 3 series of simulations. We started out just looking at what happens when only the host can evolve. And then we looked at what happens when only the pathogen evolves. And finally, we tossed it all into a coevolutionary model where both the host and the pathogen can evolve in response to each other to compare the outcome of the coevolutionary models with our other models. So looking at the host first, so here this is a raster of a whole bunch of different simulation runs. So on the x-axis, we have the maturation rate. So a higher value of m means that juveniles are maturing faster, so they're spending less time as juveniles. And on the y-axis, we have that parameter z I just mentioned, which is basically the adults' how much more resistant the adults are at baseline, where 1 means juveniles and adults have equivalent baseline resistance. So how red it is basically how much evolution is adult biased. So there's the color bar at the bottom has blue on it, and if it's blue, that means that evolution favors juvenile resistance. But basically, when we just have host evolution, adult resistance is favored across all of these parameters. It becomes even, the darker red colors mean more biased evolution towards adult resistance. And you can see as the maturation rate increases and individuals are spending less time as juveniles, then we see more evolution towards adult resistance. And as the baseline level of adult resistance increases, we see evolution is less biased towards the evolution of adult resistance. So when we look at pathogen evolution, we see, so here the x and y axes are the same, but now the blue means that the pathogen is evolving towards high adult transmission, and the red means it's evolving towards high juvenile transmission. And so as with the host, we saw kind of this somewhat gradual shift in strategy. There's this really quick bifurcation for the pathogen where it rapidly shifts from fully optimizing for juvenile infectivity to fully optimizing for adult infectivity. And this is, again, kind of given by the parameters you would expect. So as the maturation rate increases and individuals are juveniles for less time, then that means you favor trying to maximize adult infectivity and vice versa. So now what happens when we toss this into our coevolutionary model? So now on the top two panels here represent our coevolutionary model for the host genotype on the left and the pathogen genotype on the right, and below are just the figures I just showed you. So for the host, we can see that now across a broad range of parameters, we're seeing evolution favor juvenile resistance, which did not happen at all when we did not have host resistance. And the difference is less striking for the pathogen, but now we have the region where adult infectivity is favored is reduced. So what's actually going on here? So I'm going to highlight 2 points here and compare them. And so these are maturity and adult bias parameters that produce really different outcomes depending on whether or not we have coevolution. And so now I'm just going to show you what happens over evolutionary time in both of these scenarios. So on the left here, we have what's happening with coevolution, and on the right is what's happening when only the host can evolve. And so when only the host can evolve, we see that we rapidly evolve towards full adult resistance. And whereas with coevolution, initially we see the same thing happen. The host is evolving towards greater adult resistance, but the pathogen is also evolving towards greater juvenile infectivity to take advantage of the fact that the juvenile hosts are becoming more susceptible. And eventually, the pathogen becomes so good at infecting juveniles that it diverts the trajectory of the host evolution, where in the end it reaches a more intermediate final evolutionary equilibrium. So with this, our takeaways are that really both the host and the pathogen evolutionary strategy really can depend on the underlying demographic factors such as maturation rate and baseline resistance. Although, surprisingly, we see that the pathogen evolves full specialization to either adults or juveniles depending on these parameters, and there's not really any intermediate zone with this. So tying this back to transmission mode, that means at least in our setup that we're not really seeing the maintenance of multiple transmission modes when you have coevolution. And at least in this scenario, we probably need to consider something such as spatial dynamics if we want to understand why we're seeing maintenance of multiple transmission modes in this system. And so with that, I would just like to acknowledge some people, everyone in the Bruns lab at UMD who's been really helpful in this work, as well as our collaborators at UVA and Amherst College, and our funding sources. And with that, I think I have a minute or two for questions. Yeah. Yes, absolutely. And so we have some evidence for these trade-offs, at least on the pathogen side in our system, and more circumstantial evidence on the host side. It definitely does depend on the trade-offs. So lots of other people have looked at, well, maybe not lots, but Ben Ashby has a lot of papers looking at what happens when you consider trade-offs between age-specific susceptibility and all sorts of other factors like mortality. So there's a rich literature on that. And yeah, you absolutely see all sorts of different outcomes when you consider different trade-offs.
So unfortunately, I think that's our time. So before people disperse, I just want to thank all the speakers again.